Good afternoon, students. Last time we discussed about the structure of cell wall. We discussed about the various aspects related to the cell wall of the plants. Today, the topic which we're going to discuss is the cell membrane. We're going to discuss about the structure and composition of cell membrane. You should know that starting from bacteria till human beings, in all cases, cell membrane is similar in structure and its composition and even functioning. If we compare the cell wall with cell membrane, last time I even discussed about the differences. Like you should know that in case of cell wall, which we discussed last time, it is made up of like cellulose, hemicellulose, pectin, lignin, etc. But in case of the cell membrane, which is a permeable membrane, it consists of mainly proteins and lipids. Better to say phospholipids. So, and you should know that cell wall is the permeable membrane, whereas the cell membrane is selectively permeable membrane. Cell wall is rigid, multi-layered, whereas cell membrane is bi-layered and it is flexible membrane. So last time we discussed about these differences you should know. Today I will discuss in detail about the structure of cell membrane, and in this video I will specifically discuss about the models of plasma membrane, under which I will discuss two types of models of plasma membrane. That is, one is we call them as trilaminar models, and which I will discuss, one is the Danieli and Davison model, and second one is the Robertson model. The early models which were based on the structure of the plasma membrane. Before I start with the models of the plasma membrane, let me tell you a little bit about the cell membrane. Before I discuss in detail, I feel like you should read the NCERT and you should know what is mentioned in NCERT related to the cell membrane. So let's read, and then based on that we will discuss further about the cell membrane in detail. So the detailed structure of the membrane was studied only after the advent or the discovery of the electron microscope in the 1950s. So the chemical studies on cell membrane, especially on RBCs, enabled the scientists to deduce the possible structure of plasma membrane. And you should know that plasma membrane composed of lipid bilayer, which are arranged within the membrane with polar head towards the outer side and hydrophobic tails towards the inner part. I will explain you that in case of plasma membrane there are phospholipid molecules in which there is a phosphate head which is polar towards the matrix or the cytoplasm, and the non-polar fatty acid chains are inward away from the cytoplasm having water. So this is the core, is the water hating part, and the above part is the water loving part, polar and non-polar part. So that shows the amphipathic dual nature of a phospholipid molecule, which I will discuss again. First I am just reading what is mentioned in your NCERT. So here it is mentioned, this ensures that non-polar tail of saturated hydrocarbons is protected from the aqueous environment. So you should know about it. So polar heads of phospholipid molecules are outer side, whereas the hydrophobic tails are towards in the inner part of the plasma membrane. If you talk about the composition, the amount of the proteins and lipids, specifically when we talk about RBCs or erythrocytes, it's come to be 52% protein and 40% lipids. Please note down, this is very important to know about it. And then it is mentioned that in phospholipid there are different types of proteins like integral or peripheral, and some peripheral proteins are known as the tunnel proteins, which are totally buried in the membrane. So you should know about it. So here you can see there are integral proteins which are embedded throughout the plasma membrane. Some are partially embedded. We call them as integral proteins, which are present on the periphery or on the surface. We call them as extrinsic proteins. So I will show you. So here some lines are given related to the most acceptable model of the plasma membrane, to which we call fluid mosaic model. I will discuss later on about specifically about these lines. So that will be our separate that is the topic. So first let me discuss in detail about, so that was all about the NCERT. If I say from NCERT what you should know, that the extensive study was done on the RBC, plasma membrane of RBCs, and in the plasma membrane of RBCs there are 52% proteins and 40% lipids. And you should know from bacteria to human beings, in all cases the cell membrane is similar. Now you should know that the cell membrane, that term was originally used by Nageli and Kramer in 1855, and later on the same was named as plasma lemma by Plough in 1931. Plasma membrane or plasma lemma was discovered by Schwann in 1838. But later on the structure was explained by different scientists like Danieli and Davison, Robertson, and then the fluid mosaic model of plasma membrane, which was given by the Singh and Nicholson in 1972. The most acceptable model of plasma membrane is the fluid mosaic model, which I will discuss in later on this one. But now we will discuss about the structural composition of plasma membrane and the early models of plasma membrane, that is the lamellar models or the tri-lamellar models, that is Danieli and Davison model and Robertson model. So based on the study on the plasma membrane of RBCs, so cell membrane composed of phospholipid molecules and fatty acid chains. So the lipids are arranged within the membrane with the outer polar head and the inner hydrophobic tails, as I mentioned in the NCERT also. So here it is mentioned also what is the composition of the proteins and lipids in the human RBCs, and here it is explained that what type proteins are there. Now let's discuss what about the models of the plasma membrane. So we call them as lamellar models, which were the early models, but due to certain technical errors, the Danieli and Davison models were not accepted, not even the Robertson model. So let's discuss, still you should know about the history of discovery of the plasma membrane. So let's discuss about the Danieli and Davison model. So that was the first lamellar model, which was proposed by James Danieli and Davison in 1935, this model, on the basis of their physiological studies. So according to the Danieli-Davison, a biomembrane contains 4 molecular layers. So here you can see there is a one protein layer, then there is a phospholipid bilayer, and then there is again protein layer. So they explain that these are the globular alpha proteins. Here the phospholipid molecules have hydrophilic phosphate head and the hydrophobic fatty acid chains, which are non-polar. Non-polar chains are towards inner side, okay, to protect from the water or from cytoplasm, and the water loving heads are towards outer side. There is a electrostatic force between these phospholipid molecules and these proteins due to which they are held together, whereas these fatty acid chains have some van der Waals forces, kind of forces between the phospholipid molecules which help them to held together. So it means there are two protein layers of globular proteins, that is the alpha protein, which sandwich the phospholipid bilayer. It was kind of very much intact model, rigid model, because it could not explain about the flexibility of the plasma membrane. As this model could not explain that how the water loving substances will pass through the hydrophobic part, the non-polar part. So that model could not explain about this. The similar type of model was given by Robertson. David Robertson in 1959 modified the model of Danieli and Davison. If I can show you the better structure here, first look at the structure of the plasma membrane under the electron microscope. So here you can see that there is a cell membrane. Here it appears to be a bilayer structure. Okay, here again this is the diagram, electron microscopic structure. So in between here must be some fatty acid chains. So look, this model was given by the Robertson in 1959. Here it gives the trilaminar model, in which instead of the alpha proteins he proposed to have the extended beta proteins, and the protein bilayer which is towards the outside and towards the cytoplasm, they are different. Here there is a layer of glycoproteins, to which we call mucoprotein layer, and here there is a layer of beta protein which is non-glycoprotein layer, and here is the layer of phospholipid molecules bilayer, and there is a specific specifications of thickness of these layers. Like if we talk about the total length, total thickness of the plasma membrane, it is 75 angstrom, in which this protein bilayer is of 20 angstrom, this that is the protein layer is of 20 angstrom, and this phospholipid bilayer is of 35 angstrom. So 20 plus 20, 40 plus 35, 75 angstrom total thickness of the plasma membrane. We also call it as unit membrane model or the trilaminar membrane model. Again, this model was also somewhat rigid. It could not explain about the dynamic nature of the plasma membrane, that how it assists the polar molecules to pass through the non-polar bilayer, and as well as how the water loving substances pass through the plasma membrane. So these models could not explain about the such dynamic nature of the plasma membrane and the how this plasma membrane helps to pass out for the movement, for the entry of the polar substances into the cell. So that were the two early models. First one, that is the Danieli and Davison model, in which they consider the alpha globular proteins having which sandwich the phospholipid bilayer. Then the unit membrane model or the trilaminar model, it was given by Robertson, according to which towards outside there is a glycoprotein layer, inside there is a layer of beta proteins, extended proteins, and then there is a phospholipid bilayer having specified thickness like 20 angstrom, 35 angstrom, and then 20 angstrom. So total thickness of plasma membrane is somewhat 75 angstrom. So try to read these from my notes or from some reference books. So these are important one, the early models of the plasma membrane. So in next video I will discuss about the structure of fluid mosaic model that was given by Singh and Nicholson. So thank you so much.